Hello guys, the topic of this video is infectious disease epidemiology. The subject epidemiology originally developed from the study of epidemics of infectious diseases. The prevention and control of these infections needs epidemiological knowledge and experience. This video focuses on the infectious disease epidemiology. Infectious disease epidemiology is a fundamental part of the whole of epidemiology. Nowadays, we are coming through a lot many medical terms, and this video is meant for the ease to understand these terms. Also, this video will help the students to solve the multiple choice questions, short answer questions, and long answer questions in the exam. So now let's see what is the infection. The entry and then multiplication of an infectious agent in human being or in animals is known as infection. The body can respond against the infectious agent may be in the form of immune response or can be in the form of disease. So now let's see what is the contamination. When the infectious agent present on the body surface, clothes, beddings, toys, surgical items or other inanimate articles including water, milk and food is termed as contamination. What is host? When there is lodgment of an infectious agent in a person or in living animals under natural conditions, then that person or animal is known as the host. Now the question arises, what are the infectious diseases? The diseases caused due to infectious agents are called infectious diseases. Infectious diseases are usually the communicable diseases. So what are the contagious diseases? The diseases that transmitted through contacts are termed as contagious diseases, like sexually transmitted diseases and scabies. What are the communicable diseases? The diseases that are caused because of infectious agents or their toxic products. These infectious agents or their toxic products are transmitted directly or indirectly through infected person or through infected animals to a susceptible host. Now let's see what are the epidemic diseases. Epidemic is a Greek word formed from two words, epi and demos, where epi means above and demos means people. So, the rapid spread of disease to a large number of people in a given population within a short period of time. The number of cases an epidemic varies according to infectious agent, their size and type of population exposed to that specific disease. Now let's see what are endemic diseases. Endemic again is a Greek word formed by two words, en and demos, where en refers to within and demos refers to people. Thus, the word endemic stands for that infection which is constantly maintained at a baseline level in a geographical area without any external inputs. These diseases are of usual or expected frequency within a specific population. Like chickenpox is endemic, that is in a steady state in the United Kingdom, but malaria is not. Every year there are few cases of malaria reported in UK, but these do not lead to sustained transmission in the population due to the lack of susceptible vector. Now let's see what are the sporadic diseases. The word sporadic means scattered about. Thus, the cases that occurs irregularly from time to time, and these diseases are found infrequently. These diseases are very few and are widely separated in space and time, and thus the connection cannot be established between the number of diseases or cases found. Example of sporadic diseases are herpes zoster or meningococcal meningitis. Now, what are the pandemic diseases? Again, pandemic is a Greek word formed by two words, pan, which means all, and demos means people. Thus, the pandemic diseases spread across a large region, multiple continents, and worldwide. Like Spanish flu in the year 1918, H1N1 flu in the year 2009, and COVID-19, that is coronavirus, in the year 2020. Now, let's understand the chain of infection. The diseases are transmitted from the source of infection to the susceptible host through various modes of transmission. Let's discuss the term source. Basically, the source is a starting point of the occurrence of disease. Definition wise, the source is a person or animal, object or substance from which an infectious agent passes to the host. They are the immediate source of infection. A reservoir is any person or animal or plant or soil in which an infectious agent lives and multiplies. Reservoirs are primary source of survival of this infectious agent. Thus, the reservoirs is the natural habitat in which this organism metabolizes and replicates. So now you would have understood that the reservoir and sources are not always synonyms. Example, in hookworm infection, the reservoir is man, but the source of infection is the soil. The reservoirs may be of three types: human reservoir, animal reservoir, and reservoir in non-living things. Now let's talk about the human reservoir. Human reservoir can be a case or a carrier. So what is a case? Case is a person having a particular disease in a population. And carriers are infected person or infected animals that harbors as specific infectious agent in the absence of visible clinical diseases. Carriers serves as a potential source of infection for others. Carriers are more dangerous than cases because they cannot be recognized easily and continuing as they do to live a normal life among the population. Also, carrier readily infect the susceptible host. Animal reservoir. Animal reservoirs may be act as a cases or carrier. Examples: rabies or yellow fever. Reservoir in non-living things. Inanimate matter can also act as a reservoirs. Like soil may be harbor agent that causes tetanus. Now we will discuss about the mode of transmission. Diseases transmitted from reservoir or sources of infection to a susceptible individual in many different ways. The transmission depends upon infectious agent, portal entry, and the local ecological conditions. The classification of mode of transmission can be made easy by this chart: direct transmission and indirect transmission. The direct transmission includes direct contact. The infection transmitted by direct contact of skin to skin, mucosa to mucosa, or mucosa to skin of the same person or another person. This type is immediate mode of transfer of infection from source of infection to susceptible host. It do not require intermediate mode or agency to transfer the infections, like sexually transmitted diseases, skin diseases, or eye infections. The second mode of direct transmission is droplet infection. During coughing, sneezing, speaking, and spitting, the spray of droplets of saliva, nasal, nasopharyngeal secretions directly projects into the surrounding atmosphere. These expelled droplets may directly impinge upon conjunctiva, ororespiratory mucosa, or skin of a close contact. Generally, particles with diameter of 10 micromillimeter or greater than that filtered off by nose, but those whose diameter are less than 5 micromillimeter penetrate deeply and reaches the alveoli. These droplets, which may contain millions of bacteria and viruses, can be a source of infection to others. The potential of this droplet spread is increased in conditions of close contact, overcrowding, and lack of ventilation. Example, common cold or tuberculosis. The third mode of direct transmission is contact with soil. The infectious agents may come in contact exposure of susceptible tissue host like hookworm larvae or in the tetanus. The next mode of direct transmission is inoculation into skin or in the mucosa. The infectious agents directly inoculated into the susceptible host by the skin or in the mucosa. Example, rabies virus by the dog bite. The last mode of direct transmission is transplacental or the vertical transmission. The infectious agent transmitted by transplacental route. In this type of transmission, the disease agent causes malformations of the embryo by distributing its development. Example of transplacental route are torch infections and syphilis. Now, let's discuss about the indirect mode of transmission. This transmission mechanism includes 5F, that are flies, fingers, fomites, food and fluid. The requirement of indirect transmissions are the infectious agent must be capable of surviving outside the human host, that is in external environment, but they return to its basic properties of pathogenesis and virulence till it finds a new host. The character of infectious agents depend upon the influence of environmental factors like favorable temperature and humidity. There are varieties of indirect transmission, and they are: first one in the indirect transmission are vehicle-borne transmission. The infection transmitted through the agency of water, food like raw vegetables, fruits, milk, blood, serum, plasma or other biological products. Usually, water and food are most frequent vehicle of transmission as they are used by everyone. Diseases transmitted by food and water are usually alimentary tract infections like acute diarrhea or typhoid fever. Those transmitted through blood include hepatitis B. The second in indirect transmission is vector-borne transmission. The transmission by vector may be mechanical or biological. They are any living carrier that transport an infectious agent to susceptible hosts like flies, mosquito. There are various factors which influence the ability of vectors to transmit diseases, and they are host feeding preference, second, ability to transport disease agent, that is infectivity, third, ability to become infected, that is susceptibility, fourth, survival rate of vectors in environment, fifth, degree of association with man, that is domesticity, and last is suitable environment. Third mode of indirect transmission is airborne transmission. They may be transmitted through droplet nuclei and dust. Droplet nuclei are tiny particles which ranges from 1 to 10 micron and represent the dried residual of droplets. Example, diseases spread by droplet nuclei are tuberculosis, influenza or corona. A variety of infectious agents like pathogenic bacteria, viruses and fungal spores can be found in some of larger droplets which are expelled during talking, coughing, sneezing, and then they settle down by their sheer weight on floor, carpets, furniture or any objects in immediate environment and become the part of dust. During the act of sweeping, dusting and bed making, the dust is released into the air and becomes once again airborne. Like tubercle bacilli may survive in dust for considerable period under optimal conditions of temperature and moisture. Coccidioidomycosis is an example of a disease spread through airborne transmission of fungal spores. The fourth mode of indirect transmission is fomite-borne. Fomite are inanimate articles Those get contaminated by infectious discharges from a patient and capable of harboring and transferring the infectious agent to a healthy person. Fomites do not include water and food. Rather, they include soiled clothes, towels, linen, handkerchiefs, surgical dressings, etc. The diseases transmitted by fomites include diphtheria, hepatitis A, eye, and skin infections. The fifth mode of indirect transmission of diseases are unclean hands and fingers. Hands are the most common medium of transmission of pathogenic agents to food from skin, nose, and bowel. The transmission may be of direct, that is from hand to mouth, or indirect. The unclean hands and fingers denotes lack of personal hygiene, poor sanitation, which again favors person-to-person transmission, like dysentery epidemic in India in the year 1984 and coronavirus infection in the year 2020. Now we will discuss the stages of successful parasitism. The first stage is the infectious agent find the portal of entry by which it may enter the host. There are many portals of entry like respiratory tract, alimentary tract. Specifically, coronavirus find its portal of entry via respiratory tract. After taking the entry into the host body, the infectious agent must reach the appropriate tissue or the site of election in the body of host. There it finds optimal conditions for its multiplication and survival. As in coronavirus, the site of elections are alveoli in lungs. The infectious agent later find a way out of the body, that is portal of exit, so that it may reach a new host and can propagate its species, as coronavirus gets spread through coughing and sneezing. After leaving the human body, the infectious agent must survive in external environment for sufficient period till a new host is found. Now let's talk about the incubation period. Definition wise, the incubation period is the time interval between invasion by an infectious agent and appearance of the first signs and symptoms of the diseases. During incubation period, the infectious agent undergoes multiplication in the host body, and when the sufficient density of disease agent is built up in the host, the health equilibrium is disturbed and thus causes the diseases. There are certain factors which determines the incubation periods. First, generation of time of particular pathogen. Second, its infective dose. Third, the portal of entry of infectious agent and the susceptibility of an individual. Now, the length of the incubation period is characteristics of each diseases. There is a minimum incubation period for every disease before which no illness can occur. Thus, can be summarized as incubation period varies for different infectious diseases, and it also varies from person to person with the same diseases. So, why does it necessary to know the incubation period of certain diseases? They do have certain importance in epidemiological studies, like to trace the source of infection and contacts. Second, to know the period of surveillance, that is period of quarantine. Third, to advise immunization to prevent the diseases by immunoglobulins and antisera. Next, the identification of point source and to know the prognosis. If you find the video is valuable, then please do press the like button and watch the series which are interrelated, and subscribe the channel for upcoming important topics. Thank you.